Genome by Francis S. Collins and Anna D. Barker. So, cancer kills more than 1,500 Americans each day, and as the population ages, the rate is expected to rise tremendously unless we're able to find a cure or a way to attack the cells. Cancer cells have 6 abnormal capabilities to lead to their ability to spread throughout the body, and these include the following. Number 1, self-sufficiency and growth signaling, and this is where cancer cells can either amplify growth cues or even generate their own. Number 2, insensitivity to anti-growth signals. This is where cancer cells do not listen to cues from surrounding tissue to stop growing. Number 3, evasion of cell suicide. This is where usual mechanisms that normally would self-destruct damaged cells are disabled or overridden. Number 4, limitless replicative potential, and this is where cancer cells can exceed the amount of times it can divide. Number 5, sustained blood vessel growth, and here tumors can emit signals which will promote the development of new blood vessels to deliver oxygen and nutrients, which will keep the tumor stable. Lastly, invasiveness and motility, and this is where cancer cells defy the signals that would usually hold a cell in place and prevent it from traveling and spreading to other tissues. So genes code for proteins that serve as signals and regulators of other genes, and therefore a mutation might not only cause the one gene to malfunction, but it can also cause extreme damage to the cell. So you may be wondering, what are mutations? So gene mutations are permanent alterations in the DNA sequence, and these mutations can range in size, affecting only a single DNA base pair or a large segment of a chromosome to affect multiple genes. These mutations can be classified as either hereditary mutations or acquired mutations. Acquired mutations mean that they occur at some time during a person's life, and they are known as somatic mutations, while hereditary mutations imply they are inherited from a parent and are present throughout the entire person's life in every cell. These are known as germline mutations. Mutations can arise from toxins, radiation, errors when DNA is copied prior to cell division, or faulty DNA repair processes. An inherited cancer predisposing mutation being carried within a gene variant is very rare, but it can happen. Gene malfunctions are the reason that cancer cells do not have to live under the constraints of a normal cell's behavior, and the idea that changes to the genome causes cancer is not new. In 1981, actually, the first cancer-promoting gene was known as an oncogene, and since then, scientists now understand that cancer is caused by mutations in certain genes. It is also significant to note that there have been 350 cancer-related genes noted so far. First of all, this picture shows a normal gene versus a mutated gene, and there are several types of mutations that can occur, such as substitution, deletion, or insertion. Substitution is when a base pair is substituted for another base pair, usually only affecting one protein. Deletion and insertion can often affect many proteins as the entire DNA sequence shifts. Regardless of origin, though, these mutations result in uncontrolled cell replication or growth, which is characteristic of cancer. The mutations can cause the ability to invade neighboring tissues and the ability to spread to sites throughout the body, possibly disabling genes that should protect against abnormal cell behavior. Cell proliferation begins with the EGCR, epidermal growth factor receptors, in the cell membrane, and they're stimulated by growth factors outside the cell, and this will cause additional proteins and genes to grow by dividing. The article talks about certain mutations in lung and breast cancer, and members of the EGFR gene family are mutated, and this will increase the number of functions of the receptors that they encode, overstimulating this growth pathway. And changes in the BRAF gene seen in almost all melanomas also promote hyperactive cell proliferation. And once again, most cells need many alterations before they're able to become transformed into cancer cells. And ultimately, by figuring out which genes can produce this trait, can reveal which mutations cause certain cancers and therefore how to prevent the disease, which is something we'll go into greater detail in later. So Delbecco and others in the science community understood that by sequencing the human genome, they'd be able to begin to understand and eventually cure cancer. Francis S. Collins led the Human Genome Project to its completion of the human DNA sequence. So, by sequencing the human genome, they're classifying the number of genes by function and thus reveal their roles in cancer, and the next step would be to compare the DNA sequences and other characteristics of the genomes of normal cells versus cancer cells. By doing this, it can show the genetic changes that cause cancer. So many research groups have used molecular biology techniques to search for mutations in genes that are cancerous, and some recent developments, although time-consuming and expensive, show that some genetic changes in cancer cells can actually help prevent, diagnose, and treat patients. In 2001-2002, Wellcome Trust Sanger Institute began using genomic technologies, exploring cancer, and they found a gene known as BRAF, which is mutated in about 70% of malignant melanoma cases. From this, researchers tested many chemical drugs on mice to see if it could block or reduce activity of BRAF or inhibit the protein known as MEK, which is overproduced as a result of mutated BRAF. The use of technology was used greatly in the Cancer Genome Atlas, as the researchers could actually work with the idea that genetic mutations could cause cancer and find treatments from this idea. So other groups would look at genetic mutations and the best treatment options for those mutations with other severe cancers, such as leukemia, lymphoma, and colon cancer. For example, Gleevec was used to inhibit the enzyme produced by a mutant-fused version of two genes called BCR-ABL. These fused mutated genes would cause chronic myelogenous leukemia, and Gleevec works against the disease along with other rare cancers and even GI tumors. Another example is Herceptin to fight against breast cancer and the mutated HER2 gene. And several studies that found certain treatments for mutated genes led to the formation of the Cancer Genome Atlas, and this allowed great progress in treating cancer. So, less than 3 years after the Human Genome Project completion, the NIH launched the catalog of genomic changes that are involved with cancer, and this is known as the Cancer Genome Atlas. The Cancer Genome Atlas, run by Francis S. Collins and Anna D. Barker, is a way to identify all of the genetic alterations in types of cancers that gene changes can be targeted. And while Francis S. Collins led the Human Genome Project to its completion of the human DNA sequence, Barker was in charge of drug development and biotechnology research efforts with a particular focus on cancer. The article explains that this effort was not going to be easy, explaining that when applied to the 50 most common types of cancer, it could ultimately prove to be the equivalent of more than 10,000 human genome projects in terms of the sheer volume of DNA to be sequenced. So, to begin the Cancer Genome Atlas, the researchers began the same way that the Human Genome Project began, using a pilot project where protocols would be tested before the entire sequence was tested. In this situation, the pilot project in the Cancer Genome Atlas used lung cancer, glioblastomas, and ovarian cancer for the first cancers tested and matched the mutated genes. And by using these three cancers, it could be determined if other cancers could also be successfully used. So, the National Cancer Institute and the National Human Genome Research Institute devoted $100 million to compile the atlas of genomic changes for the three cancers. In this project, there's a variety of technologies that are used to look at gene alterations, such as chromosome rearrangements, changes in gene copy numbers, and epigenetic changes, which are chemical modifications of the DNA strand that will turn DNA activity on or off. There are, however, some limitations in the Cancer Genome Atlas. First, it can be difficult to separate mutations that cause cancer and mutations that are meaningless. So, in addition, mutations can mean different things for different bodies. Also, tumors have some non-malignant cells that can dilute the sample, and some mutations might be missed. So, in order to combat this problem, the Cancer Genome Atlas will link characteristics of the original tumors and outcomes of sample donors, but it is necessary that confidentiality is still held. There's also a far greater complexity in the genes that cause cancer than in DNA of normal cells, as cells are prone to a greater rate of mutation once they're cancerous. The issue of time is prominent. It could take many years of hard work and creative problem solving in order to do so. Other limitations include the question of whether or not technologies that are needed for the project can be delivered in time for the project to be economically feasible, or if they can balance rights while also developing therapies. In addition, it's significant to note that this does not encompass all of cancer research, but just the foundation. So, genetic abnormalities in the cause of cancer was suggested about 100 years ago by Theodore Boveri and his colleagues, but the idea that genetic mutations were the direct cause of cancer is becoming more popular, and here's a timeline of how that idea grew. So once again, first, 1890 to 1914, Theodore Boveri studied abnormal chromosomes during cell division and how these can play a role in malignancy. In the 1950s and 1960s, studies showed that tumor viruses injecting their genes into cells could have the possibility of causing cancer. In 1960, the first genetic defect in chronic myelogenous leukemia cells were found in the Philadelphia chromosome. In 1976, it was found that SRC, a non-viral gene in animal cells, can actually cause cancer. In 1979, P53 was found to be the most frequently mutated gene in human cancer. In 1981, HRAS is the first human oncogene discovered, and we kind of talked about it a little bit earlier, but an oncogene is a cancer-promoting gene. In 1986, actually, the first tumor suppressor gene was found, and in 1990, the Human Genome Project begins. After that, we see in 1993, Gleevec will begin its testing. So in 1999, gene activity profiles are shown for the first time, and this shows the distinguishment between cancer types and predict chemotherapy response, and this was super important for specific types of cancer in certain patients. In 2001, Gleevec will get FDA approval. In 2002-2001, Wellcome Trust Sanger Institute discovers BRAF is common to 70% of melanomas, as we talked about earlier. In 2003, researchers finished the Human Genome Project, and just 2 years later, end of 2005, beginning of 2006, the Cancer Genome Atlas was announced. And in 2006, they actually begun its pilot project. And from 2007 to 2010, the Cancer Genome Atlas collects and analyzes tumor samples from different places that are treating patients, and this is where University of Michigan Medicine will step in. So the University of Michigan has contributed tumor samples and led many Cancer Genome Atlas projects for a decade. Their goals were to understand the molecular understandings of cancer and therefore create therapeutic targets. They discovered new mutations and they wanted to understand the consequences of those tumors, and all of the projects identified subgroups of cancer which had not been recognized before, which is super significant to note. An example of this is in papillary thyroid cancer. 2 mutations that were thought to have similar responses were actually found to have different signaling properties. And this is important for Michigan Medicine because this shows the renowned researchers and their effect. According to Giordano, who has served as a professor of pathology at Michigan Medicine and as a member of Pan Cancer Atlas, he thinks that this project allows pathologists to think about cancer and re-examine the proper classification of these tumors. So, still, the issue is that the characterization of cancer is developing faster than they can create these targeted therapies, and this is a common theme in all of the cancer genome projects because it's difficult to find these genes. It takes a lot of time, just as they said it would take 10,000 more time than the Human Genome Project. So it's definitely significant to note that they need to work fast in order to combat the issue of cancer. And so the picture to the right represents the Cancer Genome Atlas run by Michigan with 33 cancer types looked at, with 10 of them being rare cancers. And this information was found based on 10,000 types of tumors, which is important because they really covered all types of tumors and all different types of patients and case scenarios. And this data was summarized with 7 different types. So here are my citations. Thank you.